So just to inform you, this webinar is being recorded. There'll be links sent afterwards to this and all the other webinars that have taken place in this series. So you'll get to watch those back. They'll be shared on YouTube. I'll also send a link out through Zoom to be able to watch back. So if you're a part of the world where you can't watch through YouTube, you should be able to watch through this. So whilst I'm waiting for people to log in, because it takes people a couple of minutes to get logged in, so we normally leave it for the first minute before really starting off. So whilst we're waiting for people to join, it's just really interesting. Could you let us know in the chat where you're joining from in the world? It's sunny southeast London here for me. Somebody else is joining in Kent. Well, I hope you're enjoying a bit of the same sunshine as we're having here. It's been absolutely appalling weather. So somebody's joining from Kent. That's great. So far, somebody else from the Kent coast. Oh, that's fascinating. Somebody else from Manchester. Yes, somebody else from East London. So we do find that this being a lunchtime session, it's quite good for people in the UK and Northern Europe. This is why it's helpful to know where you're joining from in the world. So far, it's all UK based. Interesting, very helpful. Thank you for letting us know. When we've done sessions at 9 o'clock in the morning, we found people in the Far East have joined us. So we've had people from Malaysia and Indonesia, and that's been really good fun. So I'm just going to run through how this session's going to work, how it's going to happen. My colleague Thomas will join in a moment. He'll turn his camera on in a few moments. Oh, here we go. Hello. So just to let you know how this session's going to work. First of all, introductions. My name is Kim Morgan. I'm the Outreach and Communications Coordinator for UCL Biochemical Engineering, and it's been my absolute privilege and pleasure to have my colleagues join us for these fantastic lectures, the last of which is taking place today. So it's the accumulation of a really great series for us. So some of you already found the chat function. If you're joining on a mobile device, you'll see that you've got a series of buttons, which means that you can choose how you want to see us, whether you want to have us in gallery mode or speaker mode. It's up to you how you configure the screen. Once Thomas starts sharing his screen, it'll take over the whole screen anyway. But you've also got a chat function, so if you have any problems with the sound or the visuals or any aspect of the technical presentation, let us know in the chat and we'll see that straight away. I'll also put my email address in there as well, so you can contact me afterwards, or if you have some problems with login, take note of my email address. In a moment, I'll launch a couple of polls because we're really interested to know whether you're an offer holder or whether you're an inquirer, what level of study you'd be interested in. It really helps us understand the composition of our audience, so just take a moment to fill that in. Once we've done that, I will hand over to my colleague Thomas. You can post, and you've also got a Q&A button. If you've got any question, we're looking forward to your questions. It's one of the most fun parts of the session for us. So, but you can post them at any point. You can even post them anonymously, but it's much better if you post them in the Q&A box rather than the chat. We tend to lose the questions if they're in the chat. But you can post them at any point, and then when it comes to the end of Thomas's talk, we will come to the questions. So the first thing I'm going to do is I am going to launch a poll. Let me get these up. My Zoom screen is resizing rather strangely, so bear with me. Here we go. So the first one is whether you have an offer to study with us. So we know that because we've sent out an email to offer holders, it's great when people have an offer to study degree with us. Come and join and find out some more about it. So I can see that some people have joined to have an offer, but many people don't. And as is about usual for these, most of the people don't have an offer. But if you do have an offer, it's great to have you here with us today. And yeah, thank you for coming along. Okay, that's very helpful. Cheers for that. So the majority of people don't have an offer. Interesting to know. So the other question is what level of study you potentially be looking at. So this is more for us to gauge whether you would be a prospective degree student or an MSc student. Just to make it clear, if you're a prospective degree student and any of the things in any of the aspects of this webinar, if you're not sure about the science or you're not sure about it, don't worry. If you come and study with us, what we do is we get you to that level. So so don't worry about it. But I haven't had anything back on that yet. Bear with me a moment. Sorry, that was a bit of an issue with that one. launch this one. Oh, sorry. Apologies. Sorry, Zoom is really, really being strange at the moment. Yeah, there we go. Fantastic. So that's the Zoom is allowing me to ask you this. So yes, this. So it seems that most of the people who are joining us today are prospective, or more at degree level. We've got a few postgraduate, which is typical for these sessions, and it's entirely how we design them. But just to reassure you, our modules will take you to the level where you would understand all of this completely. So that's great. Thank you very much for completing those. What I'm going to do now is I'm going to hand over to my colleague Thomas, who'll introduce himself and go through the lecture. If I turn my screen, when I turn my screen off, I will be here in the background. So if you have any problems, you can just use the chat function and get straight through to me. But Thomas, over to you.
Hi, so can you all see my screen? Is that all okay?
Yep, that looks great. Sound coming through good as well.
Excellent. So just to echo what Kim was saying, I've decided to kind of pitch the talk I'm going to give you very, very early, very basic to begin with, and then it gets progressively more and more in depth. When I started my BSc, I didn't know any of this. I finished my PhD, and I've done all of this. So it gives you the whole gamut of where we go. So my talk's going to be on innovations in autoimmunity, because that's the field that I work in. Just very quickly, there we go. I'm going to set this to laser pointer so that we can see where I am. Okay, so very quickly, a little about me and my background. I did my undergrad at the University of Hertfordshire, doing a BSc in biomedical sciences. I then did a placement year at the Health Protection Agency doing virology. I did free fetal genetics. That's where the entire genome of the baby exists in the circulation of the mother at Great Ormond Street afterwards. I then did my PhD, which I'm going to talk to you about. And that was kind of an MRC case thing. So it's between industry and academia. Then I moved on to doing a postdoc in immunology, a fellowship in biophysics, which is where I came across biochemical engineering, really. And now I work in antigen discovery. The point of this is not really to say, look at all my stuff, look at what I've done, but people will often show you their careers in this format where you have your successful criteria underneath. So big numbers, 3.5 million, lots of ticks, lots of novel, lots of new, and how you go through. If you're looking to be a scientist, bear in mind, this is more the way the career looks. So you hit a lot of roadblocks, you go to a lot of different places. So often your career won't look as linear as people will present. I tend to do this couple of slides whenever I speak to anyone who is coming into science, because I remember when I was at your stage, people always presented and I was like, God, they've done everything so linearly, it's gone so well for them. It doesn't go that well for anyone. It's just how it's presented. So like for myself, for example, post PhD, I ended up in immunology, but I'd failed to get into there once and tried to get into biophysics, couldn't make that. Got into immunology, and then from there went to the biophysics stuff, didn't get into industry at this point, and then have found myself background into where I wanted to be eventually. So you do get. Now on to the stuff I'm going to talk to you about today. I work on a disease called antiphospholipid syndrome. It's reasonably rare. It's 5 in 100,000 people. It's got a very odd and difficult diagnosis. So it focuses around the presence of antibodies. These antibodies, just up here, have a Fab region, which binds to antigen, and an FC region, which binds to a cell. This can potentiate any number of sort of different problems in the body. This is an autoimmune disorder. So in healthy people, you don't get these antibodies. In the APS people, or people with antiphospholipid syndrome, this is what generates the disease. For diagnosing these people, we have laboratory variables. So these antibodies to, this is called beta-2 glycoprotein 1. This is what I'll be talking a lot about. So you have anti-beta-2 antibodies and then these others. Clinical variables for this disease, you get thrombosis. So this could be, for example, a PE, so down in the leg, a DVT down in the leg here, PE up in the lung, or a stroke, or obstetrics. So you get things like miscarriages and intrauterine growth restriction. It's actually the leading cause of strokes in people underneath 50, and it's the leading acquired cause of miscarriage. So how do we treat it is the question. And the answer is imperfectly, as it is with most things. So we have one treatment, which is called warfarin. This is a non-specific vitamin K coagulation blocker. This is kind of just global prevention of clotting. So Your coagulation system works using vitamin K as a cofactor, so it needs it to activate, and this just binds all of the vitamin K and prevents any clotting. The very obvious problem then is if you do cut yourself as normal, you will continue to bleed. So we're not just dealing with the disease, we're now creating a different problem. It's reasonably effective at preventing recurrent clotting, but it does have problems. You can't use it during pregnancy. You can't reverse it. So if someone's in an accident, it's very hard to maintain them afterwards. Doesn't work in all patients and has quite a significant problem for dietary habits. You can't eat leafy greens. You can't have, your spinaches and things because you'll put more vitamin K in and it will cause a problem. One of the alternatives we have is called heparin. Heparin is used during pregnancy. It's much easier to reverse, but it requires daily injections. So this is obviously bad for patients. It works the same way with the vitamin K, so you're still at the same risk of bleeding. So it's a bad disease and we need better treatments. So if we were to design a new therapy, what would we do? Well, it would need to be at least as effective as what we have. We want to get better. We don't want to get worse. And it would need to be used long-term in asymptomatic patients. Now, this is an important point to make. With APS, people get treated after they've had a symptom. So you've had a stroke, you've had a miscarriage, you've had a DVT. Some people identified that have the antibody, but they've yet to have the outcome. Now, those people are called asymptomatic. And at the moment, the treatment is kind of almost as bad as the disease in some ways. You couldn't give someone warfarin or heparin for life because of that. You can give them other things that are less effective, but we need a new therapy. We're going to design a therapy that means we can actually treat those people. We can try and prevent them having their effect. Needs to have fewer side effects than anticoagulation. We need to let people eat what they want and be able to, cut and bleed normally and heal rather than overbleeding. And the way you do that is something called targeted action. So at the moment, we just give these coagulation blockers, and it's very, it's a carpet sort of approach. It attacks everything. What we need to be is more specific. We need to design something for this disease, which is specific and is going to have an effect. So we know the antibodies cause the disease. Why don't we target the antibodies? Why don't we do something that stops those antibodies? To do that, we need to understand more about the main antigen, the thing the antibodies bind in APS, which is beta-2. So this is beta-2 glycoprotein 1. Studying this has been about the last 10 years of my life, so I'm very familiar with this. It's a 5-domain protein, so you've got 1, 2, 3, 4, and 5. 5 is quite different. You see these big old loops down the bottom. That's what lets you bind to cell surfaces. And then domain 1 up there is the main antigenic target. It has roles in complement and coagulation. So coagulation is just blood clotting. Complement is my favorite part of the immune system. What it does is it allows you to punch holes in cells. Basically, it's the end point of immunity. It's a whole lecture in itself that I won't go into, but it's a very interesting set of molecules. As I said, domain 5 down the bottom here binds anaerobic phospholipids. And we have multiple antibodies to this. So I've already said about IgG antibodies. Those are the ones I showed in the diagram at the start. You also have things called IgA. So Ig just means immunoglobulin. That means antibody, basically. And IgG are the long-lasting ones that we usually find in the blood. They're the ones that most assays look for. IgA is more in your mucal membranes, so in your stomach, in your intestines. There is some in your blood, there is some in your mouth. What's interesting is that the antibodies for both of these types, cause disease when you have beta-2 like this. So that really says that this is the thing we need to target. If you can get multiple antibodies with one drug, that's really what you're looking for. And as I said, domain 1 is the main site of antibody binding. They correlate best with thrombosis. So what sort of therapeutic potential does this molecule have? Well, if this is needed to bind to the cells down here, and this binds to the antibodies, what happens if we just produce this piece? We don't bother with the bit that can create the, can bind to the cells. So we don't have the disease potentiate. So this is basically what we started doing. So we produced, this is just domain 1. So this is that top piece that was in red. And we started trying to produce it. The question is, can we target the antibodies? This is what I like to call a decoy peptide. So this little orange bit here is where the antibodies bind. That whole molecule, that's the piece that's the problem. So if we put in something like this that's soluble, that can't bind to cells, the antibodies will bind this and then will be dealt with normally. They won't bind to cells and cause disease. So in theory, you're creating a decoy for the immune system. The problem is this is incredibly small, even for a protein. Its half-life would be measured in minutes. So you'd put in 100 micrograms, within 5 minutes, 50 would have been gone. So it's really hard to have a drug that you can give to someone that's active, that has that sort of shorter half-life. The reason we've got to inject heparin is the same thing, because it very quickly gets taken out. So we need to modify this. We can't just make this and put this in. It's not suitable for a therapeutic. Now, there's a process called pegylation. It's the binding of a polyethylene glycol, or PEG moiety. All of you have come across PEG at some point. It's in every hand cream, it's in every face cream, it's in tons of things. And the reasons it's in tons of things is because it's inert, so it doesn't react with things. It's inactive, it doesn't have an active phase that it does, and it's non-immunogenic. That makes it pretty much perfect for throwing on the side of a protein to make it bigger and try and help with the elimination. The problem is when you try and bind two things together, how do you control where it goes? I mean, you could have it anywhere on this molecule. It could be anywhere on the surface. So what we did was something called targeted conjugation. What this does is, in yellow here, we have a disulfide bond, and down there we've got another one. This is more yellow because it's more surface exposed. So we know we can target that, and we should get our PEG bind to it every time. And the thing is, you need a balanced half-life, so how quickly you lose the drug versus how active the drug is. So if we put in this non-pegylated form into the body, it would bind antibodies really well. It would be really quick, and it would fully bind as many antibodies as it can, and it would be gone in a couple of hours. Your body will produce more antibodies. It's not going to have a long-lasting effect. However, if it stays in there for, say, a week, it's got more opportunity to bind antibodies, but it may not be as efficient because you've put a big protein, a big PEG moiety on the side. So maybe it loses 50% efficiency. Well, that's fine. It's there 100 times longer. So you recover that activity over time. A lot of drugs do that. There's something called pegintron, which has 7% of its affinity, but because it stays around long enough, you end up with it doing the active portion that it needs to. So can we inhibit these antibodies causing the disease by making this drug? Just very quickly about PEG, polyethylene glycol. We say it increases the hydrodynamic size through a hydrated backbone. All that means is it gets bigger, and it's very good at binding water. It acts upon proteolysis. This is just enzymatic breakdown. So your serum is full of enzymes. Those enzymes will come across proteins and we'll just cleave them, because that's what enzymes do. When you do that, you can start to lose effectivity of a drug, because it's no longer the drug it was. It's now broken into pieces. Renal clearance. If you imagine your kidneys are just a bit of mesh, like a sieve, the bigger your protein, the harder it is to pass through the sieve, the more you'll keep in solution. So PEG is very good for increasing the size of that. And then the last one is immunogenicity. And this is actually just based on, if we go back one slide, the surface area it hides. So the bigger the PEG, the more of the surface area you can't see. If you can't see the surface, you can't form an antibody to it. The irony is we're already in a situation where we want to bind antibodies. So that's actually less relevant for us. As I said, there's a conjugation problem. There's also a size problem. So the bigger the PEG, the more of the protein you shield. The longer it stays around, but the lower the activity is in theory. So this is that sort of balance that we're looking at. So let's produce the drug. Day one of my PhD. This is the vector that we use to produce the drug. So it's just into an E. coli cell, and in bacterial cells, you can put in little vectors with something called a lac operon, which is here. And that lac operon will mean you can add in something called IPTG, which is like an analog of lactose. Put that in, and it will force expression of whatever's next to it. So you now have a switch. You have a switch you can access inside a bacterial cell. What we have here is the bit that we want to express. So we've got our 6-histidine tag, our AVI tag, and then this D1 is what we're after. So if that whole of that whole vector is only that little green bit that we want to produce. All of the rest of it helps in other ways. This CAN-R gives some sort of antibiotic resistance. So when you grow your bacteria, you can put in a little bit of kanamycin, which is what that gives resistance to. Only the cells that contain your vector and therefore the potential to express your protein will survive. Everything else will get killed. So it's a way to control what's going on. So we can put this into cells, we can put it into media, and then we can see if it expresses. So this is how you look at an expression. Each of these are different media. So what you can do is you can take your bacteria, put it into a solution, and then give it different nutrients to see, how it responds. Different things will grow different proteins in different efficiencies, should we say. What we're looking for is where I've got this little band called band of interest. That little band there is what we want to see. The brighter the band, the more the protein, the happier we are. So in this top left one, we've got all different media. The last one, T6 here, seems to be the one with the biggest blue band. I hope you agree. That's TB. That stands for terrific broth, and that's very well named because it's very good at what it does. So we know that terrific broth is our best media. If we move across to the right, what I should mention is actually that I've put, we've produced two different versions of this protein. One of them works very well. One of them is abandoned halfway through drug production, which is what you tend to do. You find your lead candidate and continue. Up here on this one, we have different inductions. So you put your bacteria in, you put your kanamycin, you now have a bunch of bacteria. You allow that to grow. The more bacterial cells you have, the more protein you can produce. However, when you switch your switch on and you say produce the protein, you're going to be in a situation where you're going to start exhausting the media. It's going to start using all of the glucose, all of the nutrients to produce your protein. And the length of time which you allow it to do that is important, because if you do it for too long, it will start to degrade itself and destroy the protein to try and make more protein. So this is just a look in saying, what's the right sort of time? And you can see the strongest band is in number 7 and number 13, both of which over here are 16-hour inductions. Fantastic. So now we know we've got TB at 16-hour induction. The next thing to look at is temperature. So when you induce, so when you grow a cell, 37 degrees for E. coli is perfect. It's the correct temperature because it's used to human growth, it's used to human temperatures, fine. When you want to grow a protein, sometimes lower temperatures are more stable. So what you do is you test 20 versus 37, and here, this is just a densitometry graph, or an intensity graph. So this is just measuring how blue that blue band is. And you can see pretty well the yellow bar is the best bar. So that's TB, 16 hours, 20 degrees. The last ones over here are all very similar. These are very small losses or gains. So you can switch your switch on harder or softer, and you can switch your switch on earlier or later, basically. So the first ones with the IPTG, increasing concentrations, harder switch turning on. The red bar doesn't really change. The optical density, this is how many cells you have when you start producing. So this is 5 is some, 6 is a lot more, 7 is maximum. And you can again see you don't get a huge difference. So because we've optimized those first 3 steps, the media, the induction time, and the temperature, these other ones aren't having as much of an effect. So we can go for the lowest IPTG, which is the cheapest option and the best environmental option. IPTG is quite toxic. And then we can go for the highest optical density, just to give us the greatest chance of getting protein. So at this point, and I'm aware we are going quite quickly and there's a lot being thrown at you, again, I wouldn't expect everyone to understand everything that's going on. I didn't when I started doing it. You'll pick it up, as Kim said, when you come over and start doing these sort of things in projects. So now we want that blue band, but it's in this mix of everything else. So we have to purify it, right? So this is a chromatogram. This is one of the ways we can do large-scale purifications. And if you remember, when I showed you the vector, I said we've got 2 tags. One said 6-his, one said AVI. The point of that is 6-his is non-natural. It doesn't exist in nature. So you can create a column which will bind to that unnatural thing. That means everything that's normally in a cell should flow through your column, and then you'll be retaining just what you want. And that's what we see in this. So the blue line here is a measure of protein. This is me putting the lysate in. So this is just the raw stuff from this lane, or technically this lane, I think, going through. And what you'll notice is there's not really much around where we want. And that's because it's bound onto the column. So at this point, everything that you don't want goes through the column. And then as it comes out, you have this peak here where you've changed the pH. So for histidines, you deprotonate the ring, and that allows it to fall off a column. So you drop the pH to like 4, and you get this. This is all of the protein of interest that we want. Interestingly, these little bits are called laddering. When you get a lot of protein, it starts to just associate with itself in solution and as it goes through the gel. So these are all evenly, perfectly spaced to be like 2 of our protein of interest, 3 of, 4 of, multiples as it goes up. Okay, so we've produced the non-pegylated form. That's great news. What do we have to do next? We have to pegylate. Now, I'm not going to go through the chemistry of pegylation for 2 reasons. One, it's proprietary, so I got apologies. It's proprietary, so I can't tell you because the company that I worked with would be most upset. But I will give you a few things that have been published in a second. But what you need to know is, you remember I said we can target those yellow bits, those 2 disulfides, one at the top, one at the bottom. That means our maximum amount of PEG is 2. That's dipegylated, 2 pegs to 1 protein. What we want is monopeg, 1 peg, 1 protein. That's because dipegylated, in theory, with the 2 molecules, will shield more of the protein. So you'll lose more activity because you can see less of the surface. And we want to bind antibodies, right? So we can create monopegylated, which is what this is in the middle here. And then this is the unconjugated material. Now, one of the fun things about this is that it's a proper conjugation reaction. So you add in a reagent, and the more reagent you add, it can go across towards, dipegylated, but then you can slow the whole reaction down by putting it at colder temperatures, like 4 degrees, or you can activate certain parts of the molecule. So it's a full chemical conjugation process that I'm not going to force you guys to listen to now. But yeah, so the aim is to get this monoconjugated. And you can see that's where the strongest blue bands are. That's where the highest blue peak is. So we've got a pretty good method. Yeah, that's the molecule we want. So this is what I can tell you about the conjugation. We worked with a company called Abzena. They make antibody drug conjugates, or what used to be called silver bullet therapies, where you have an antibody with a toxin on the end that it creates. It basically allows you to target tumors, but you can use their conjugation methods for other things. So we use this thio bridge, targets disulfides, thiols specifically, with PEG on the end. And we use this in the top right corner here. So these are all the different methodologies they use. I'm aware this feels a little bit like an advert for Abzena as it stands. Apologies for that. But this is our rebridged version. Now, it's important it's rebridged. When you have a disulfide, you break it, you create 2 thiols, 2 SH groups, right? You can then undergo a nucleophilic attack with your PEG reagent, which will bind to one. The binding to one side will then kick off the leaving group of the other, and it will bridge. And it's specifically designed to be the same width of a disulfide, because these things are like staples for proteins. If you break them, the protein will begin to drift apart. It will begin to change its structure. So you want to maintain that structure as much as possible, especially if you're doing antibody binding, because antibodies recognize a surface. As soon as you start to change that surface, it's a problem. If you think of it like a lock and a key, if you have the right key for the right lock, great. But if you then start to screw around in the lock with like a screwdriver or anything, you start to damage the barrel, it won't fit properly anymore. So it won't do its job. So you want your disulfide to maintain its proper structure, and that's why we use this reagent. Just a very quick look at purity. So this is something called reverse phase HPLC. This just binds protein and releases it according to its hydrophobicity, so how well it binds to long chain hydrocarbons. What you're looking for is sharp, tall peaks, and only one of them in an ideal world. And what we see here is one big sharp peak. So there's pretty pure protein with a little peak off the side. In the red box here is what we've put in. So it could be there's a little impurity or there's something slightly off, but it's about 95% pure, which is biologically pure. And that's plenty for what we were doing. Now, in 3 and 4 are bigger sizes of PEG. So we've made variants to see whether we can use a bigger PEG or a smaller PEG. So there's it. We've produced the drug. We've made the drug. How do we know it works? This is where we get into the immunology side of things. So there are things called ELISAs. For an ELISA, you take a kind of plastic well like this. You put your protein, that's beta-2, with the purple bit being domain 1. And then you can put patient serum in and detect the binding of antibodies. It's just raw antibody detection. If you wanted to test a drug in this situation, what you can do is before you put the serum in, you pre-incubate it with your drug. Here we're calling it recombinant domain 1, and it will bind to some of those antibodies. This means those antibodies are no longer capable of binding to the thing you have on the plastic, so you see a drop in signal. So you start to see no inhibitor very high, and then you see, as you increase inhibitor, you see a drop in the signal. And that's exactly what we see. So these are 6 patients with APS that we took their blood and we tested the drug in it. And basically, as you increase the concentration of the drug at the bottom here, you see the curves generally go down. So we have wild type PEG and non-PEG, and then our other mutant type of PEG and non-PEG. What's quite interesting is the patients in the blue box. The non-PEG works much better. The PEG works okay, not as well. You've lost a bit of activity, but as we said, you expect that with PEGylation. You see again here, the same with this patient, APS 12 and APS 14. You see the same sort of drop. But these 3 saw absolutely no drop in their signal. So they have pretty much identical curves to the PEG and the non-PEG. So this suggests even within APS patients, specifically within the IgG portion, you're going to get patient variability. We know that their antibodies are going to bind slightly different places. You do the same thing with the IgA. If you remember, we talked about the antibody that's more in the gut and more in the mucosal membranes, a little bit in the blood, still causes the disease. You see the same thing. You see inhibition. So it goes down. It's a little less clean than the IgA, the IgG. The signal is a little more wobbly, but you see, again, the same sort of inhibition. And we ended up doing that over about 20 patients in each group. And when you average it out, you see your non-inhibitor has a lot of binding, and then you're looking at about 50% loss of inhibition, a lot of loss of binding. So 50% inhibition for your IgG. Oddly, with the IgA, you see increased loss of binding. So you see better inhibition in the pegylated forms. It may be that because it's more stable, or it may be something to do with the structure once you put the PEG on. But either way, this is great news for patients. We now know we can inhibit patient binding to patient antibody binding. So you retain over 90% of activity. The problem is, this is just very non-physiological. You've got a piece of plastic with a protein on it, and then you're putting blood on it, and then you're putting in your molecule. It doesn't really reflect what happens in the patients, So I developed a new coagulation assay. So this is looking at how the patient's blood clots. So there's a thing called the lupus anticoagulant, which is used for diagnosis of APS. And basically, it measures the amount of time it takes to clot once you put in a reagent which starts the clotting. It's actually snake venom. You add snake venom to human blood, and you can measure the speed in which time the blood clots. You fire a laser through it. The laser has a detector the other side. And as the laser gets blocked by the formation of a clot, it will give you the speed at which that happens. What I found was you can take purified antibodies from patients. So these are the things that potentiate disease. Put it into the serum, put it through the assay, and you get the extension you expect. So then we can do it in the presence and absence of the antibody, of the drug that we've produced. And what you see is, as you add the drug, you get a drop. Everything above 1.1 here is considered positive. So all these people are positive for APS, and they have clotting problems. And you can reverse it. So you put in the non-PEG, it comes down. And then you put in your other 2 PEG sizes, and it keeps coming down. This is likely because, you remember I said that PEG helps prevent proteolysis, so it stops the enzymes breaking the protein up. That's probably what we're seeing here. It's sitting for an hour at 37 degrees. This one is likely getting cleaved. This one is likely having some sort of more stabilized effect through proteolysis. So now we know it works in a kind of a clotting environment. The last thing I'm going to talk to you about with this is a mouse model. And I just want to reassure you before we go through this, it's very humane. It's done through every ethics board that we can get hold of. and we do it in the minimum approach. The whole reason we produce this coagulation test is to reduce the amount of animal testing that goes on. So we have an acute model where mice are given IgG from patients and then are given the inhibitor an hour later, and then you measure thrombosis 3 days later. And then you have a chronic model where mice are given beta-2 glycoprotein 1, they produce their own antibodies, and you give the inhibitor at the end. And what you see is a significant reduction in thrombosis in both models. So here we have our blue, which is APS IgG. So that's our maximum signal. That's when you put patient antibodies in. And this dark blue is our inhibitor. So you get a significant drop. Now, for the pegylated form, it's not quite as significant. It's not as good because of that loss of activity we see. But what we see in the chronic model is a retention, if not a slight improvement, with the 40 kilodalton peg. This is the effect of half-life. So for the acute, the molecule is only in there for about an hour before you start testing, right? But the chronic, they've developed their own antibodies and you've given them this hours before. So this effect is fine, but they're producing more antibodies the whole time. So that's why we see the peg has improved the ability of the drug. And that's kind of the final outcome is clotting. But we know if you measure certain things on cells, you can see how well are they starting to clot in advance. And this is measuring something called tissue factor, which is one of the ways that we can generate clots. And this is actually on monocytes in the intraperitoneal space. So it's somewhere we can get to quite easily. And what you see is in APS alone, in their IgG, you get tons of production of tissue factor. This is how they're creating the clot, they're producing these factors. And then in our drug groups here, you don't see production of it as much. It's all back down. And then it's the same here with the chronic model, where they've got a pretty high level here, not as high as the acute, but still pretty high. And you get that drop, and again, the biggest drop with 40 kilodalton peg. So it works in binding, it works in coagulation, and it works in mouse models. It's currently going through production with a company to get into first-in-man trials in a couple of years. But we're still being reactive. We're not being proactive. So these people are still having to have clots before we start picking them up. So can we diagnose better? And this is some fairly obscure biophysics I thought I'd include just in case some of you are very interested in biophysics or are mathematically minded. I said to you before, beta-2 had a J shape, and we looked at it, had the red at the top. Well, it turns out you can modify it. There's something called plasmin cleavage that happens in patients. And what that does is it cuts the very end and the top flops over. Why is that relevant? Because it changes the way that the epitope is. That bit that stuck out, remember the orange at the top of the molecule that I said this is where the antibodies bind? That changes. And we can tell that changes because when you do the ELISAs and you try and pick up inhibition, you get significantly more inhibition from plasmin-clipped beta-2. What's fun about that is plasmin is activated during clotting. So it's sort of a feedback loop. But the other thing we can do now, is we can start to look for the structures themselves in patients. Why did they develop the antibody in the first place? You know, we're still treating an antibody that produces, but it would be better to prevent them getting the antibody. So this is a thing called ion mobility mass spectrometry, which is one of the methods that we have over in biochemical engineering and structural molecular biology, which is insane. Basically, you have a field of ions. So this is just a charged area, and you can throw proteins through it in a cycle. And as it goes through, it will separate the structures of the protein based on the surface charge. It's absolutely mad that we can do this. And we know beta-2 has a circular and a linear form. The circular form, you see that red where the antibodies bind, it's kind of hidden by this green, but it's very open here. So the question is, do APS patients develop antibodies because they have more of the one on the right and less of the one on the left? Very simply. So down the bottom on the left-hand side, we have the healthy control. This is where we have our little bump out. That's fine. APS, some APS patients are like this, where we have, again, a small amount of the extended. So the later, sorry, I should say the later peak, this peak is the extended. It's this one on the right. But then you get some APS patients where you have more extended and some where you have almost only extended. So this is brand new research that we've really only got together in the past month and a half. Maybe a little longer, but not much more. It's starting to get to the point where we've begun with a disease, We've then looked at the treatments, we've made a better treatment, and now we're trying to find how we can prevent them needing the better treatment. How can we get earlier and earlier in the process? And this is only possible when you combine immunology with biochemical engineering. Because to design the drug, we've used a lot of the principles of biochemical engineering. And to do this sort of assay, you've used the biophysical engineering. So it's why I like to jump between fields, and it's why I like working in biochemical engineering at UCL, because someone will talk to you about a method and then you'll come across something cool like this. So just to finish off, this is me back when I had hair, where the day we got the patent, because we patented the drug. These are the other guys that worked on it before me. And these are all the people that have given me funding over time. And that is the end of my seminar. At the moment, I'm open to questions and answers as and how. So Kim, do you want to manage the next part?
Hello, yes. So just whilst Thomas is having a look through, in fact, if you, we're looking forward to your questions. Could you put them in the Q&A box? I see we've got one already, but you can post them anonymously. Anything you'd like to know any more deep, about any of the aspects of the presentation, please pop them in the Q&A box. Whilst I give you a chance to put those together, I just want to mention a couple of things that we've got coming up. The first is that if you're interested in studying a degree with us, or any degree at UCL, we've got a series of online virtual open days happening later this month. Myself and Dr. Jack Jeffries will be online on the 23rd and 24th. One late in the day, one early in the day, so you can come along and find out more about studying a degree with us. I'll put a link to that in the chat as well, so you can follow that up. If you don't have a chance to open it now, it's fine. Go to the front page of our website, simply Google UCL Biochemical Engineering. You'll see both those events are listed there. We also have an in-person open day spread over 2 days, and that will be on the 28th and 29th of June. This is the ideal situation for us to show you around our unique facilities. We're the only university in the UK to have a pilot plant. It means that we can actually show you the unique facilities, introduce you to the team. You can meet the academics, you can meet the students as well. It's really a chance for you to find out what it's like in our department. But if you can't make it in person, or you'd like to speak to us first, as I said, we will be online later in this month. Right, I can see now that we've got a few questions that have come through. Just to reiterate, Thomas, we may not, and to the people who attended, we will do our best to get through them, but if we don't get a chance, we'll do our best to answer them all. Thomas, do you want to answer? I mean, if you want to answer them in order, or pick whichever one you think you want to go for first.
I'll start working my way through. So someone has asked, does PEG act like a shield around the protein molecule? Absolutely. You get two types of PEG. You get linear and you get branched. The more branched it is, the more it's like a globule that will sit round. You get some where it will extend and then wrap, but we got quite lucky. We didn't have to do too much for that. Will the drug be able to be used on any clotting condition? It's just APS specific. The theory can be used on other conditions, any antibody-based condition. For things like protein C deficiency, we actually produce protein C, or you can produce activated protein C and modify it that way. So it's a slightly different approach. How do you discover or find and model the structures of proteins? So there's a thing called the protein database, PDB. If you put anything you're interested in PDB, the crystal structure is probably already published. If it's not, there's a really cool thing that UCL are very heavily involved in, especially in biochemical engineering, called AlphaFold, which allows you to predict structures. And there's an awful lot, especially within Professor Dolby's group, who kind of just resolve these structures. It's fantastic, but I couldn't possibly tell you how AlphaFold works. Did my degree in biomedical science open many doors, not just autoimmunity? Yeah, I've worked in genetics, virology, drug development, biophysics. Your BSc should be seen as like a jumping off point. It's where you go to learn the basics and then learn what you're interested in. And why did I switch from lupus to APS? Well, APS, my parents had, my family have, so it's pretty easy to skip across to that. Since then, I've gone across to things like RA, Sjogren's. I don't believe in siloing to single diseases, and it's something that biochemical engineering actually taught me, was techniques are translatable. So things you learn should move to new fields. That's where you'll find new and interesting things. So I switched to APS because of the family, but then I've switched to every disease I can get my hands on since. um, what are the mechanisms of autoimmunity? Oh, good Lord. So what mechanisms cause autoimmunity? The classical three are things called molecular mimicry. So you get a disease like Epstein-Barr virus, and one of the proteins from that looks vaguely like a human protein, and then you end up generating antibodies that cross-react. The other thing is you break tolerance, there's a lot of it, or it looks different, and your body doesn't have the right signals to say, okay, that's self, don't attack it. So it produces an antibody. Or, there are a dozen other ones that we couldn't possibly start to get into. How can you stop people producing antibodies in the first place? So we have things like rituximab, which delete B cells, so it depletes them all. So the idea is you get rid of them, and then you don't have those B cells. Hopefully, you don't reproduce them. We use that for something called ITP very regularly. The other thing people are doing now is CAR T cell therapy, which is way above my grade. Basically, they're training the immune system to target the immune system in a specific way to get rid of them. And they are using it in APS. There is a trial in lupus that's going very well. It really is probably going to be the next wave of drugs. And then probably the last question, are optoisomers enzymes taken into consideration for creating this drug? So fantastic question. I have no idea where you're from, but if you have an offer, I hope that you do come along. So we do have a racemic mix, basically, because we have the two potential binding sites. So you have your kind of, I call it the proximal and the distal disulfides. We don't know if there is a predominance to one or the other, because we'd have to do a lot of purification to do that. Ultimately, we don't overly care at this point. So we're trying to produce a drug. We know it works. We know it's reproducible. It's really for the next phase when it goes to like a GSK, where they then have to answer all those FDA, like the Federal Drug Association questions, and have release criteria. That's their problem, not my problem. Thankfully, it's a really good question. That's it's quite in depth.
Absolutely, thank you. We've got a few moments if there are any more questions come in, but just before we wrap up, I also want to say that we've got UCL Engineering is actually 150 years old this year. It's an incredible achievement to get that far, but we're doing a festival of engineering across the whole of the faculty. And bearing in mind that our faculty of engineering is bigger than a lot of universities, and it covers a wider range of subjects, obviously biochemical engineering, chemical engineering, mechanical engineering, security and crime science. And we'll be showcasing not just what we've done in the past, but also really looking to the future. So if you'd like to follow us on social media, do check that out. If you're able to come into London this summer, we've got a whole week of really exciting stuff going on. Some of my colleagues will be giving talks, some of them will be doing presentations, but it's a great chance to find out how we're linking together engineering across different disciplines to try and tackle global issues. I think hopefully this last talk has really illustrated how engineering and biochemical engineering particularly, but engineering generally it really is a multidisciplinary solution to solve global problems. So as Thomas mentioned, if you're interested in the CAR T cell therapy work that we're doing, there will be something from our colleague Pedro Couto, who did a talk earlier this week. So you can find out more about what we're doing in that field as well. In fact, if you're interested, we do an MSc in manufacturing commercialization of stem cell and gene therapies. Officially the longest program title at UCL. We've got a Q&A session coming up for that next week as well. So yeah, I think we've got through all the questions, which is a testament to you, Thomas, to get through those. But they were really great. Thank you for that. That's been it's been really great to have those. If there's anything you want to follow up, I have put my email in the chat, but I'll also be contacting you in the coming weeks as well with links to the recordings. But it just comes to say thank you very much for joining us today. It's been great to have you with us. Huge thank you to Thomas for doing the last of these talks, and I really hope to see some of you in the future at UCL in person. Have a great day.
Bye.
Thank you very much.